Hello, I'm just going to give folks a minute to get into the webinar before we start. Just generally takes a couple minutes to get started. All right. Hey guys. We'll go ahead and get started, and hopefully folks will keep rolling in here as we go. Um, Hi, welcome. I'm Darla Clayton. I'm the Community Engagement Specialist for the Alliance to Cure. And I am excited to welcome Andrew Ressler tonight. Um, Andrew is a postdoctoral researcher at Duke University. He's been working on CCM research for 2 years. Andrew published a paper on a genetic mechanism that leads to the growth of lesions in Nature Communications last year. He started simple science sessions to communicate directly with patients about his science and general scientific questions. Andrew's PhD was in stem cell and mouse models of rare genetic epilepsies. I just want to mention real quick that Andrew isn't a medical doctor, so we're going to keep our questions on the science side, not the medical side. And speaking of questions, we will have time for those, and there is a Q&A feature. So I would appreciate it if you could use the Q&A. I may not see questions if you put them in the chat, so using that Q&A will help get your question asked. And I also wanted to mention that we have a support group that will kick off right after this meeting is over, so right around 8 o'clock. And without further ado, I'm going to pass it over to Andrew. Thanks, Andrew.
Thank you, Darla. So everyone, today I'm going to do a little bit of a double conversation. First, I'm going to talk about the simple science sessions that I started to talk directly with patients. And then I'm going to talk a little bit about some of the basic science behind CCMs. And today, what I decided to talk about was what really makes them different from like a cancerous tumor. So I talk about cells, I talk about how these grow and how they grow differently than cancer. But before that, I'm going to talk about something that I've been doing lately, which is a way to communicate directly with patients. And as I go through this, there's going to be a couple times where I stop and it's going to be a weird silence for like 15 seconds because I ask a question. I know no one's going to be able to answer it in that short period of time, but I like to keep people engaged because I want to make sure that when I communicate science, I give people a chance to think about what comes next, because then I feel like everyone's more engaged and people hopefully will learn more. So, and then at the end, I'm going to try to move a little bit quick through some of the slides, and there's going to be a lot of time for questions. So I like to answer questions more than I like to give a presentation, because I feel like then I can actually talk about what's most interesting to people, but you got to give a presentation first. So we'll start there and hopefully I don't bore you. So first, what are these simple science sessions? I went to the CCM International Conference and I really liked engaging with patients. And one of the big parts about that was actually being able to hear patients' stories. And I think for scientists in rare disease, it's actually really important to learn from the community, because when you study something that millions of people have or are experiencing, you don't really learn that much from an individual. When you're studying something where it's not particularly well known, you guys might know this from your experience going to doctors or talking to people, you might say CCM and the person might say, I've never heard of that. And when you have a disease like that, that really is rare, this community is really important, but for the scientists, we can actually learn a lot more from you guys as well. So part of this was for scientists to learn from the patient community. And then the other part was for us to kind of practice talking about what we do or a scientific concept that you're interested in directly to patients. And the initial approach of this, which I did, and then it's on hiatus right now. I'm going to try to start these back up in a couple months. And if people are interested, please, I'll give you an email to talk about it at the end. But the initial approach was to have two 15-minute conversations. And this is going to change a little bit because we want to get a few more patients in each of these so that we can talk to more patients. So we're trying to figure out, is 3 or 4, like how can we do this so it's still a big back and forth? Because I really wanted these to be kind of a conversation more than a talk. So the way that we did this for the initial pilot experiment was we did two 15-minute conversations. The first one was just here on the left, the tell me your CCM story, where the patient who wanted to do this would just say whatever they wanted. You can share as much or as little about your CCM story and your life in general as you want. but we try to get an understanding of what you're most interested in. Going forward, we might do that with an intake form or something, but it's pretty critical that we kind of know what type of science you're interested in hearing about, and or if there was a specific scientific paper that you read that you wanted to hear about, or if you just wanted to hear what someone was doing. And these conversations had a wide variety of topics. Some of them were about genetics and the role of a gene called PIK3CA. Some of them were like how bleeds in these CCNs can lead to nerve damage, and what mouse models and studies in patients tell us about that. One was about honey and brain health because someone was interested in that comparison because they saw on a podcast or something. So we actually dug into it because here there's, as scientists, you can always look into something. You never know what's going to be interesting. And then one was on spinal cord cavernous malformations, which are less common, but a researcher at UCSD, Helios, studies these a lot. So he was talking about these. And then one was on the use of focused ultrasound, which came from Delaney's work. So a few different scientists helped me with this, so thank you to them. And when we did this, the conversations were intended to be about 15 to 20 minutes long. And the most important part here is that people tended to like these and want to have more conversations. So of all the patients, all of them said they wanted to have more conversations. Almost all of them wanted to have more conversations with the same scientists. And that was something I was really excited about because people get to know each other. And I think when you get to know each other, people get more comfortable asking questions and you tend to learn more from each other. And then they also wanted to talk to other scientists and hear what they were doing. The other thing was patients felt that the information was delivered at an appropriate level of complexity. And this is something that you can all tell me whether or not I do a good job of this at the end. It's harder when there's more people to make sure because everyone has different scientific strengths and scientific weaknesses. Everyone has things they know more about and they know less about. But we really try to make sure that when we talk about a scientific concept, people understand. And one of the things that someone told me when I was a scientist, and I do want feedback on this always, and was that if you can't explain something to someone who knows nothing about what you're doing, it means that you don't understand it that well. And I really believe that. So if I start talking and you're like, oh, this is a neuroscientist, he must be so smart, that means I'm doing a bad job. If you're like, oh, this actually seems kind of easy to do and study, that means I'm doing a much better job. It means I know what I'm doing. So when I have these conversations, I really think it's important that I try to describe them in a way that people can really follow and learn and become more advocates for themselves and what they know about what's happening in their own bodies. The other thing is patients left these conversations feeling a little bit more hopeful. And this didn't always mean they were more hopeful for themselves, but they might be hopeful for the next generation. And as you all know, science can move slow and it can move fast. It can be many years where there's no real scientific advancement. And then in a year or two, everything can radically change. So it's the same thing with this. So a lot of the science we talk about, some of the science will be stuff that might be relevant 20 years from now. Some of the science might be relevant 5 years from now, some of it 1 year. So when we talk about this, we really like to talk about scientific concepts. And when we talk about this hopefulness, I'm not someone who can tell you what to do clinically. If you want to know what drug is better for you, I'm not the person to ask. I'm not a medical doctor. But what I can tell you is what's happening behind the scenes, what's coming up, what we know, what we don't know, and what we're trying to find out. And sometimes when you hear about all those things, it starts to paint a picture of a field that really is on the verge of delivering therapies and improved lives to patients. The other thing was the anecdotal responses. So patients, I just asked them to kind of say what was the most valuable and what recommendations do you have. And people mostly said do more of this, but they also emphasized hope, promise, and learning. And a lot of it was hope and knowledge. I think I do believe that knowledge is power, and I think knowing more about what's going on can make everything a little bit less scary. So hopefully these conversations can also understand, like, make it so that people can understand what's happening. And I think for some people that can be valuable. For some people, it's not very valuable. For some people, it doesn't really do anything, and that's okay too, because we'll learn from you and we'll keep trying. So that was that. And now that's kind of the background. And if people are interested in having these conversations going forward, I'll give you an email at the end. You can reach out, and we're going to hopefully start up these conversations again at broader scales. Hopefully, maybe 4 or 5 people at once and one scientist. And we're going to try to start that up again either at the end of this year or early next year. So if you're interested, I'll give you an email address at the end, and you can reach out. Now I'm going to start talking a little bit slower because we're going to talk about some science. Today, today, what I am focusing on is endothelial cells and how do they lead to lesions. So now I've said a word. I've said endothelial cells. What do these mean? Most importantly, what an endothelial cell is here, I hope you can see my cursor, but here, these are the endothelial cells, and this is the anatomy of a blood vessel. So in every blood vessel in your brain, you have these endothelial cells right here. And on this side, where it says the lumen, that's where you have blood. So you have a bunch of blood in here. And then you have stuff that grabs onto the endothelial cells. And then you have your brain. Here's your brain. And here's another image of it. You have your endothelial cells here. You've got your blood underneath it. And then you've got your brain. So what does it look like these endothelial cells are doing? It looks like they're creating a barrier. And that's what they do. The most important thing about this type of cell, and as we talk more, you're going to understand why I focus on this cell and not all the other cells in this figure. There's all sorts of cells in your brain. You've got everything here with all sorts of different names. But I'm only really talking about this one, and you'll figure out why in a few slides. But the first thing is, what do you think these are doing? And here you see there's these little gaps right here. They've got these terms here, but in like Claudin and Occludin. But there's basically these gaps right here. What's happening in these gaps? Maybe stuff is going out and stuff is going in. And that's what they do. They create a barrier. And when this barrier is working properly, your blood releases nutrients to the cells that need it, releases some oxygen, other different anything that's in your blood, any sort of nutrient or lipids, which are fats or acids that your brain needs to function well, can leak through here. And then also in the reverse, anything that needs to get into your bloodstream can come in through here. So if you're trying to do some signaling, so if your brain's trying to tell somewhere else that something's going on, it might release a chemical into the blood, go through the blood to some other area, and then release that back out. And that's how they can talk to each other. So it's also a way for the brain to communicate with cells further away through the blood. So this is really tightly regulated. It's really important that these endothelial cells create the right type of barrier and the right types of things can get through and the wrong types of things can't get through. So what's actually going on here? So this is from a study where you actually zoom in and you look at the different types of junctions that endothelial cells have. So what a junction is just how the two endothelial cells connect to each other. So here's a graphic from the previous slide, but this is what it actually looks like. So this on the left in figure A, that looks more like an actual blood vessel. And what they're zooming in on here is that in these blood vessels, you actually have these thick cells and they directly hold on to each other. These are called adherens junctions. And all that means is that they're basically like a little bit of like sticky note or glue on the outside of each cell so that they kind of glue to each other. And these adherens junctions are really, really important to creating a very firm barrier. Nothing really gets through them. They stick, nothing moves. So these are really important for making sure nothing goes in or out. Okay, great, they're good stoppers. But sometimes we want to let things in. We can't just, if this was completely like, if this was all adherens junctions, there would be nothing that got in or out of the blood vessels. And then you wouldn't be able to actually give nutrients to your brain and your brain wouldn't be able to signal to other people. So you have this second type of junction called a tight junction. And these two interact with each other. But these tight junctions are a little bit different. These tight junctions are also ways to connect, but it's not like a sticky note. It's more like a handhold. And those handholds kind of like move in and out a little bit. So they can create little gaps for things to come in and out. And there's all sorts of different processes that will tell these cells when to loosen their junctions and when to tighten them. So there's all sorts of signaling that tells these endothelial cells that are grabbed onto each other, okay, let's open up a little bit and let this through, or let's stay closed and let's not let it through. And then you can see here, I've highlighted one of the genes involved because it's a CCM gene. This is CCM1. So this is one of 4 genes that we know directly cause cerebral cavernous malformation. So we know that this is one of the causes. And this is really important in creating these adherens junctions, but also it's important in regulating the tight junctions. So you have all these CCM genes, they regulate both of these junctions. And when you have mutant CCM genes, then these junctions are not functioning properly, and all of a sudden things can go out or in. So what do these and their junctions have to do with me? I already talked a little bit about the CCM genes, but these CCM lesions, which you can see here, this is just a picture of one. I'm sure you guys have all seen much better pictures than this, but this is just a picture of one. I like it because it's like a big glob. This is not how it is actually going to look in your brain, but it makes it very easy to talk about. So this giant thing right here is a CCM lesion. And it's basically a blood vessel that forms incorrectly. You don't see this perfect, like kind of like cylindrical circle blood vessel. It looks kind of like it looks more like a blob, and it's got kind of like, some people will say that it look like popcorn shaped, is how some doctors and scientists will describe them. But what happens is when you don't have these junctions, you start to get these weird looking blood vessels. And sometimes people call these vascular tumors. And this is what I'm going to start talking about. And I know that Darla had told me sometimes when you guys communicate, you'll refer to them as vascular tumors. And tumors, obviously, that sounds like cancer. So what is a vascular tumor versus a cancer? And that's something I'm about to talk about. But the reason why they call them cancers is because just like cancer, these vascular tumors or lesions or whatever word that you tend to use to talk about these CCMs, they're chunks of cells that are not doing their job well, and they're forming wrong. And before I get to what's the big difference, what happens when these form wrong? Two things are basically happening. Either blood leaks through because the junctions aren't tight enough, and then now they're just a little bit too open, so blood is coming out of the blood vessel and going on your brain, and brain bleeds can make you sick in all sorts of ways, or they're just not actually transferring blood how they should. So that can end up more like that's what creates like the risk of stroke, or it just doesn't get the proper nutrients to the places they need to go, and that can cause a wide variety of problems. And finally, just kind of like cancer, if it's big, it can just be like this big clump of cells that's not doing its job and makes it harder for other cells to do their job because they're putting pressure. So wherever this is, cells that should be doing their normal brain jobs can't be because it takes up space. So what's different? They're both CCMs and cancer are these big malformed blobs of cells that aren't doing their jobs correctly and that are putting pressure on cells that need to do their jobs correctly. So what's the real difference? And now we're going back to endothelial cells, unsurprisingly. So it's endothelial cells in a proportion of 6 cells. So before I talk about this, I'm going to ask a question. So here in the top left, what I'm showing you here is from a paper from the lab that I work in that was published about like 6 years ago by a great scientist named Matthew Detter. And what he did is, and I'm going to walk you through this experiment real quick. So what he did here is in the top left, you see all these like genes. This is called a construct. All this basically is how we are mutating the mice. So what you're seeing here is pictures of mice. So we're trying to mutate the mice, and we're giving them these CCM mutations, like CCM mutations that some of you might have. We get the same mutation that we've seen in a patient, and we give it to a mouse. But when we give it to a mouse, we're not giving it to all their cells. We don't do that. Instead, when we give it to a mouse, we're only giving them to this, and I have this in quotations, to the PDGFB cells. Okay, PDGFB is a gene, and it's only in certain cells. And now I'm going to be silent for about 15 seconds when I ask this question. What cells do you think PDGFB is in? There's a huge clue. If you had to guess, what cells are going to be the PDGFB cells? Okay, the answer is endothelial cells. So it goes back to that. So this gene is only expressed in endothelial cells. So when we do this mutation in the mouse, and this is one of the things we can do as scientists, we can decide which cells we put a mutation in. We're not going to put it in all your cells. We're only going to put it in your endothelial cells that make the blood vessel. And what happens when we did that? What happens when we did the mutation, but we only mutated cells that were endothelial cells? The answer is they got lots of lesions. So here is a mouse brain. Each of these are lesions here. So that's a lesion, that's a lesion, that's a lesion. So what does this tell you? And this tells you that you don't need to mutate all the cells. If you only mutate the endothelial cells, which, just quick reminder, the endothelial cells, they're the inside of your blood vessels that create these tight junctions that make sure whether or not blood goes in or out. But when I only mutate those cells, I get these lesions. So we know from that in a mouse, if you mutate endothelial cells alone, you get the disease. And then we've also done these kind of studies where this is some of the work that I actually do in my lab, where someone has a CCM surgically removed, and then we look at that. And when we look at the surgically removed cells, we actually find that the mutated cells, again, are the endothelial cells. So these are the cells that, when they're sick, create these lesions forming. And what they do is they make it so the blood vessels can't form properly and that these tight junctions don't work as cleanly as they want. So that's one real difference, because when you think of cancer, breast cancer, that's going to be like tissue in your breast. If you think of colon cancer, that's going to be cells in your colon. So one of the things is this is specific to endothelial cells. So that's what makes it different from a lot of different cancers. But you could have a cancer of an endothelial cell. You can have a cancer of any cell type. So what makes this different than a cancer that would have endothelial cells? Now we're going to the proportion of sick cells, which is where we have these fancy colors. And I'm going to make these colors get bigger again. So this is actually pretty cool. This is a technique that was created like 15 years ago that I find really interesting. And it's basically called confetti. And you have like these colors that is actually named confetti. So these are called confetti mice. And what this means is it means they mutate cells different colors. So like when we actually look at the brains of these mice, we can see which cells came from the same initial population. So these are all cells that came from an initial population, and these cells are sick cells. So what we can basically do with this, zooming everything out, what are you looking at? Each of these colorful dots is a sick cell. And then anything that's not colorful is a healthy cell. So these colorful dots are the sick endothelial cells. And the other dots and the other no colors, which is most of what you're looking at here, are basically healthy cells. So when you look at this, you say, oh, I can really see these clean dots. It seems like I can really see these cells. And when you see this, as someone who's done a lot of staining, anything this clear is not in that high percentage of cells, especially in 3 dimensions. And these are 3-dimensional images. So this is slicing a bit of the mouse brain and then looking at these. So this is a really small proportion of cells. Most of the cells in these lesions are healthy. It's only a very small number of cells in these lesions that are sick. So basically, it's a small number of endothelial sick cells that alone leads to the growth of these lesions. So then I'm going to ask a question again, be silent again, and then I'm going to show you, what do you think this image would look like if instead of these colors, these are just 3 different lesions that are different colors, but it's the same concept each time. What do you think this would look like if this was cancer instead of a CCM? Because I said the proportion of sick cells are a lot lower with CCMs. What do you think you're going to see when I show you this exact kind of confetti mouse stained for cancer? Five more seconds, I turn the page. Okay, so this is what you see on the right. So cancer is on the right, and that kind of looks like just a very colorful blob. And the reason why is you can't actually even see single dots because they overlap with each other so strongly. What you're actually seeing here is this is 2 different tumors because they mutated a lot. They mutated a lot of cells. So this is 2 different tumors in a mouse, and it was a confetti mouse. So it's the same type of mouse, the same type of staining. And with the CCM, you see discrete dots. You can actually differentiate the mutant cells, and you can see that they're not touching each other. They're kind of distributed throughout the lesion. When you look at this in cancer, it just kind of looks like a blob. And there's different ways. So they'll do these studies where they'll look at cancer over time, and early on, when the first cancer mutation happens and the tumor starts to develop, you'll get images that look kind of like a CCM, where it's only a few sick cells and there's lots of healthy cells. But overall, these cancer mutations are just much more powerful. They overwhelm the system. So they basically just take over the entire population of cells. They just grow and grow, and they take over the nearby healthy cells. Whereas these CCM lesions, you don't have them having it so that all of the cells are mutant. Instead, you have a very small number of cells that are mutant. And for a scientist, this actually makes our job often a lot harder, because when I'm actually looking for something, it's more of a needle in a haystack than just looking for the hay in the haystack. In cancer, every cell you touch is a sick cell. In a CCM, there's so many healthy cells and so few sick cells that when you try to study it, you have to be really careful to make sure that you're actually looking at the sick cells and you're not just looking at healthy cells that don't really tell you as much. Okay, so what's in one of these CCM lesions? So this is the same basic concept, but what does this actually look like? So if you like, and this has happened, some people when they have to have surgery, a surgery removes the CCM, then it gets sent to us, and then we try to see what's actually in this. So this, let's say like we have a CCM that gets sent to the lab, we try to look what's into it, and then we find a way, which isn't easy, but let's say we find a way to separate the healthy endothelial cells here, which is the light blue, the mutant endothelial cells. So these are the sick cells. And then you also have blood cells and then other cells that are in your blood vessels. There's so many different cells in here. But when I actually get a lesion and I'm trying to look for sick cells, oftentimes I can't see the cells as clearly. Instead, what I'll do is I'll get like 100 data points. So I'll say like, okay, tell me what the gene is in 100 cells in this lesion, but I won't know what I'm looking at. I just know that it's 100 cells. So if I get 100 cells, how many of them are going to actually be sick? It might be here it's 5. And that's kind of the number that we often see, where I'll get a lesion, I'll do genetic sequencing, and then I'll be like, okay, how many sick cells are actually in here? And the number will oftentimes be somewhere between like 1 to 5, right? So it'll look like this. You got some sick endothelial cells, you got some healthy endothelial cells, and then you got lots of other cells. So this lesion is really, like the word we use in science, is heterogeneous. It's got lots of different cell types in it, doing lots of different things, but the one that's causing the problem is just this small number. So it's a small number of things causing a problem. And what would this look like in cancer? Very different. In cancer, let's say the healthy cells are these white little healthy cells, and then the sick cells are these red ones. This is what a cancer cell would look like. This is what a tumor would look like if we actually had a tumor removed and sent to our lab and we sequence it. And when we sequence that, honestly, almost everything we sequence is going to be mutant. There's going to be some healthy cells, but most of the time, most of the things I see are going to be cancerous cells. So these lead to like very different ways of studying it. So when I study a CCM lesion, I'm trying to optimize my methods to focus on a small number of cells. Whereas when you're studying cancer, so that like, and just let me go back 1 second. What does that actually mean as a scientist? If it's a small number of cells, how is that going to actually change what I do? And the big explanation I have for that is a lot of my work is focused on determining what's real and what's noise. Because when you look at things, there's always a little bit of noise. And for a CCM lesion, because there's so few mutant cells, if I see something that I'm like, oh, this seems like it could be contributing to disease, I have to make sure that it's not just a little bit of random noise. Whereas in cancer, you don't have that problem the same way, because if you see something, you'll see it in 80% of the cells. You'll see it all over. Whereas here, I'll see like tiny little dots. And I'm like, okay, are those tiny little dots really what I think they are? Or are these just random little dots? So you just kind of have to really focus. So some of the things I'll do is say, okay, can we actually take the endothelial cells out of this lesion only, and then just look at those cells? And that's what a lot of people do. And you learn a lot by just looking at the endothelial cells. Now, what some other people will do is like, they'll say, and one of the things I'm really interested in now is what's actually happening to these wild type endothelial cells. Because why is it that even though there's so many healthy cells, they can't have the junctions that they're supposed to? Is there something happening in the wild type cells because they're close to the sick cells? So then that's another question that I'll ask. Is there something different about the wild type cells? And you don't really ask that with cancer. So it's very different questions. And those questions can really have an impact on what kind of therapies could work. For instance, let's say that we can figure out a way to kill these sick cells, right? Are there enough healthy cells that that's just going to lead to a normal blood vessel now? If I can kill these, does everything get fixed? Or what if instead of killing these, I make more of the healthy cells? Would that fix it? Or is just the small number of the sick cells going to hurt me? Whereas in cancer, that's not going to help. If I kill all these, there's nothing left to do its job. Whereas here, there might be enough stuff to do their job that if I could just kill some of the sick cells, the healthy cells would be able to take over. So that's a lot of the questions that we're asking as scientists. And that's kind of all I have for today. So the last thing is just going back to the first thing that we talked about. What happens when you have a sick endothelial cell? Why does it cause a problem? And that's here going back to the same image. So I'm just highlighting, let's say all these other ones are healthy and this one's sick. And then here in that green and the blue, those are the tight junctions. Now, all of a sudden, the junctions might not be tight enough. Blood can leak out, the wrong things can leak in, and just one sick cell, even though it's surrounded by healthy ones, can break the whole system. Okay, and why don't these grow? Why don't these grow like cancer? And the answer to that is sometimes they do grow, sometimes they don't grow. Typically, they grow very slowly because these sick endothelial cells divide, but they don't divide nearly as fast as cancer cells. That's why they only take up a very small percentage of the lesion. They're not that much more powerful than the wild type cells. They're a little more powerful, but not a lot more powerful. Whereas in cancer, the mutant cells are way more powerful than the healthy cells, and they just take over. Here, they don't have that much of the scientific term again is competitive advantage. They don't have that much of an advantage, so they're not going to just take over. And this also leads us to a bunch of questions. Why don't they take over? Is there a way that we can learn from that? So what is different between these and cancer? And can we push that even further to ensure that they don't grow? Because some of these will grow and we don't really know why. So one of the things that we're doing in my lab now is I'm getting lesions that grew bigger and lesions that didn't grow as much. And I'm trying to figure out, why do some of these behave a little bit more like cancer and grow a little bit? And why do other ones just stay the same size or even shrink? Some of them shrink and we don't really know why. But if we could see why, if we knew why some of these were shrinking, maybe we could figure out the mechanism behind that and encourage them to shrink as well. And then the other thing here, just another big difference, is these are not contagious. Cancer, if one of those little cells comes into the bloodstream, goes somewhere down the bloodstream, and then ends up somewhere else in your body, it can grow a new tumor somewhere else. That's not how this works. These are only in this spot. So the lesion itself may grow, but like one lesion is not going to chip off, go somewhere else in your body and cause another lesion, or somewhere else in your brain. That's not how it happens. So these lesions will develop, they might grow, they might shrink, but they're not going to be contagious. They're not going to start creating lesions all over your brain. Whereas in cancer, it can cut off, show up somewhere else in your body through the bloodstream, and then create some cancer over there. So that's all I have for you. Now let's do some questions. And then here, I'll leave this here. If you have any questions or you're showing interest in the future, if you just provide me with your email for simple science sessions in the future with other scientists talking about their work or questions that you're interested in, send an email to this, like announcing your interest. So in a few months, I'll keep track of who's ever interested when these get back up and running. Okay, now I'm ready for questions.
Awesome. Thanks, Andrew. That was super interesting, and I felt it was very understandable. I hope everyone else did too. All right, I have some questions that came in beforehand, so I'm gonna hit a couple of those, and then just remember, I know I saw a couple of questions in the chat. I will attempt to grab those, but if you could use the Q&A, that would be super helpful. All right, so questions that came in beforehand. I have one question. I'm going to try to make it a little more succinct, but essentially they have, this person has the familial CCM2 gene. They have lots of people in their family affected, and she's trying to get a gauge for how come certain generations seem to have no symptoms and other people seem to be having symptoms younger. And, you know, is there a reason that we understand for why that might be happening?
Okay, so there's two potential questions here. There's one, which is, what is the actual, like, how does inheritance work? What's the genetics of that? So why do some people get the gene and some people don't? Is that part of it? So I'll answer both. And so for that one, in that question, the way it basically works is every gene in your body has two copies, one from your mom, one from your dad. When you get this disease, it means you have one bad copy of the gene and the other one is okay. Okay, so that means, like, if I have a kid, I have two copies, but I only give one of mine to my kid. So if I give the healthy copy, my kid won't get sick. If I give the mutated gene copy, then my kid will get sick. So it's a 50-50 chance for each child that they might get the mutation or they might not. So that's why some people might get sick and some people might not have CCMs at all. Now, the second question is, for why is it for some people they get way more sick than other people? And what that comes down to is, it goes back to one of those early slides where I said, we don't know why some of these lesions grow and why some of them stay the same size, why some of these lesions sometimes even shrink. And the reason is each lesion behaves differently. And that's because the lesion is not created just from that mutation. It's created from that mutation and other stuff that happens as that cell becomes a blood vessel. There's a bunch of different things that are interacting. So each lesion is basically its own little person almost. Like, it has its own genetics, it's got its own information. So every lesion, even so Darla, she's as you guys know, she's a patient with a familial one, so she's got several lesions. Each of the lesions she has has their own complete different world. And if I looked at them, I would find something new about each one. And some of those worlds are not that bad. And some of those worlds for people's lesions are really bad, and those grow more or more aggressive or bleed more. So each one is its own world. So each lesion you have to kind of think of as its own thing. It's not like you just get one disease. No, you get a bunch of lesions, and each lesion has its different behavior. And oftentimes when people get really sick, it's a single very bad lesion. And that's why surgery is recommended for some people, because they'll take out that one really bad lesion and all the other lesions are not really causing a problem. For some people, multiple lesions are all causing problems, but each lesion is its own world. If that doesn't answer the question, you can ask it again and then let me know.
I think it did. All right. So the next question I think kind of ties into that, which is I'm just going to read the whole thing. It's a little bit long. This person has CCM1 and they have lots of cavernomas. They started to have problems when they were 15 years old with headaches.
Mhm.
And obviously they have the genetic type, so all of the cells in their body will be affected. So they're trying to understand when they had the second hit to change things from a passive to an active condition, or are they thinking about it wrong?
Okay, so they're thinking about it right. So for other people, this concept of a second hit is basically what happens is you have, I told you, one sick copy, one healthy copy, but you don't have a lesion in every blood vessel. You have plenty of healthy blood vessels. And then sometimes you get these CCMs. Why is that happening? It's because really early on in one of the early divisions, so you start from like a single zygote, then you have a blastocyst, which is like 6 cells, right? And then each of those cells, every time they divide, you might get another mutation. And most of those mutations are completely fine. We evolved to have mutations because mutations are usually good. Most mutations are neutral or they can have some sort of benefit. It's really important for everyone to have mutations. The problem here is you already have one sick copy of CCM. If you get a second sick copy, that's when the lesions form. That's when it's really bad. But it's random. So each time a cell divides, there's a small chance, like a 1 in 10 million chance that it has a mutation that's bad. And if that bad mutation happens to be in the CCM gene, now that cell is going to turn into a lesion. So that's what's happening. And typically, almost always, it's not always the case, but almost always, this happens in utero. So these cells are dividing. So in utero means like as you're a fetus, as you're developing before you're born, each of these cells are dividing. And then something happens where it gets the second hit. Now it turns into a lesion. And those lesions tend to be there when you're born or really early on in your life when cells are dividing a lot. As you get older, you're not really making new blood vessels that often, but sometimes you are. So the vast majority of the time, these are coming from birth or early childhood, these events. So you have the second mutation really early on in your life, and then you might just not notice it for a long time, or it might grow slowly. So all of a sudden it looked really small, but now this one over many years has grown and now causes a problem. But usually they started there really young. Every once in a while, and this is typical, like CCM sometimes will emerge after radiation therapy. What radiation therapy does, like if someone has cancer, like after radiation therapy, they're starting to see these CCMs and I've started to study them. And basically what happens with radiation therapy is when you do radiation, you destroy some blood vessels so they have to grow back. And also you create some DNA instability. Radiation induces mutations. So now you have a situation, it's almost like when you were born again, where you now have blood vessels growing, mutations happening, and then you can actually get a new lesion in adulthood. That can also happen after a stroke or trauma or injury or anything else that destroys blood vessels, because once the blood vessels have to regrow themselves, you're now at risk for getting that second mutation.
Thank you. That was very understandable also. Okay, so the next question might not be totally in your wheelhouse, but I'm going to throw it out there and you can answer it or pass. So this one is that they've heard a lot of different things about what people with CCMs should or shouldn't do. Things like don't do blood thinners or aspirin, don't use probiotics, or maybe they can actually help. Should they be avoiding emulsifiers or not? Is things like scuba diving, roller coasters, high elevation, should people, is there any science that we know about those things in terms of their safety for CCM? What are
Yeah.
your
So first thing, not a medical doctor. talk to your doctors about these type of things. So I'm going to talk a little bit about like the science and things that we know that can affect things, but I'm not gonna mention specific like roller coaster or things like that. If you want to talk about a specific activity, I saw that, like I was also like reading one of the notes. I saw that there's a med student here who has questions who I'll follow up with offline. But talk to someone with a medical degree about specific activities. But what I will say is, in general, these CCMs respond similarly to other blood vessel issues. And when I'm talking about that, I'm talking about people who might have like obesity or different types of things, or like any sort of blood vessel disease or heart disease and stuff like that. And what that basically means is most of the things that your blood vessels need to be healthy, you want them to be healthy for CCM as well. So if you have low oxygen can be really bad for your blood vessels and your cells. Like low oxygen is going to be bad for CCMs. If you have high levels of stress, for instance, stress is known to really hurt your blood vessels. So if you have high levels of stress, it'll hurt your blood vessels. And anything that hurts your blood vessels in CCM, you're just a little bit more fragile. So diet and exercise, it's the same kind of thing. All of the stuff that's really important for maintaining blood vessels in general tends to overlap, and all of the like bad things that tend to be like, there's certain things that are triggers in a dish. So like, let's say we cause hypoxia, which is a lack of oxygen, the CCMs grow more, right? Let's say we give the mice a high fat diet, which we've done. We give the mice a really high fat diet because sometimes we have a mouse model and they don't create lesions, and we don't like that. So we're like, because you can't study anything if there's nothing there. So then we're like, okay, why is this mouse not having lesions? You switch them to a really high fat diet, and then all that fat in the brain and stuff will actually make these lesions grow. So there is a lot of evidence that typically things that are mean to blood vessels for regular people are just meaner to blood vessels if you have a CCM. So that's not really helpful, because if someone told me, like, especially like if someone's like, oh, just be healthy, like I'd be like, well, that's a rude thing to say. Like, I don't know what to really do with that information. So I don't like, but there are, for like certain people, and doctors will have good ideas of things that you can do, just like people will do it with they have a different type of heart disease or blood vessel disease. They'll give you recommendations. There might be specific things that are even worse, but generally, anything that makes your blood vessels unhappy is going to make your CCMs unhappy.
Thank you. That was a great answer. All right, so I'm going to hit a couple questions from the chat.
Yeah, so I'm looking at those. So I was going to answer. So like the last one, I just saw this, gut health. So gut health is, because that kind of tied into what I was just saying. I saw this, and the answer to that is a very firm yes. And the problem is, going from the proof of principle in science to actually using this is really hard. But what they actually did was, we had these mice that we would give CCM lesions to, and they had lesions all the time. And then that same mouse strain, so they had the same mutations, and then they sent like one of those mice over to us in a different lab, and those mice never had lesions again. They didn't grow CCMs. And it took us like 3 years. So this was before I was in the lab. I joined the lab as they were figuring this out. And what they basically ended up learning is that there was a very different distribution of bacteria in one lab than the other. And this wasn't in the food. The food was the same, but just in the environments that they were in. The mouse, like, basically you create these mouse apartments and you just have like this whole mouse room. They all have very different bacteria in them. And they realized that the different bacteria in the gut actually determined whether or not these got lesions. And if you messed up the gut, the lesions would grow. So they actually did that. They actually basically put these like animals, they basically did like the reverse of an antibiotic, which means like we just added more bacteria. And when we added more bacteria, the ones started to grow lesions again. And then we took away all the bacteria and just basically cleaned out the entire gut and perfectly controlled what was in their gut. The lesions went away. Now, taking that, so there's a lot of researchers who talk about gut health, but when it comes to gut health, it's a full environment. It's not like there's one bacteria that's good or bad. If there was, that would be amazing. Then we could just take that away or give it. But the problem is, our lives are really, really complicated, and controlling a human's gut health when they're moving around doing things, like if I put you in a cage, you never got to leave, and in that entire life, I perfectly controlled your environment, maybe I could control your gut. But any person who's walking around, controlling your gut is harder. So people are looking into that. But the basic answer is, gut health does matter, but how will that actually work with humans and exactly what about the gut health matters, we still don't know. But it's a really good question, and it's really interesting because everyone was really surprised by that. It took like a couple years to figure it out, but gut health really did matter. And that's true with a lot of diseases. The brain gut axis is something very interesting. Okay, so the one above,
It's
if at
interesting.
61, I got if at 61 I got diagnosed with too many, so I'm reading this so people know. If at 61 I got diagnosed with familial CCM with too many to count in my brain after first symptomatic bleed, if I got thrown to concrete ending up in hospital a month later due to speech issue that began after the attack, is the treatment plan different than if it had sporadically bled? So this is where again the doctors will know the treatment plans and like what to do, but in general we do not have a good enough understanding to really tailor treatment plans perfectly to someone's genetics and the type of thing they have. It is different with familial just because you have more stuff going on. So doctors will treat you differently and do different things if it's familial versus sporadic. But in the future, when we know more and like some of the stuff we're doing now is for the first time, there's a neurosurgeon who removes a lot of these lesions, and he's removed some from familial patients, from sporadic patients, and we have really good information on the phenotypes that they have. So we know how much they bled, how big they are, and we track them over the years. And this data has taken a while to get. And now we're starting to try to tie the genetics, so what specific familial mutation you have, how many lesions you have, how big they are, to the phenotype. So we know the mutation, we know how it's causing you. And that's the first step because we have to actually have the information, both in terms of what the genetics are and what your symptoms are. And we need enough of those. So we're doing this with 100 lesions. So there's 100 different patients. And we're hoping that that's going to give us good information. So that in the future, doctors will have more information and more data to make these decisions. Because right now, doctors are going to do this very much based on you, the symptoms that you have, the experience you have. When they look at the scans and they see how many lesions you have, where they are, how they're doing, they're going to talk to their teams and they have all sorts of like ways to then make the right decisions to treat you as best as they can. But from a scientific perspective, there's no hard and fast rule based on what genetics you have. And we're trying to figure out exactly how a different genetic mutation or familial and sporadic, how those really behave differently to then give the doctors the ability to really do this not just by symptoms, but also based on biology.
All right. I wanted to go back over to the Q&A, and there was a question about sporadic versus genetic. So in sporadic cases, researchers hypothesize that PIK3CA mutations from the developmental venous anomaly is the main driver for
Mm-hmm.
the first and second hits. And they're wondering, how do you imagine the second hit occurs in familial cases?
Okay, yeah. So, um, okay, so this, um, I'm going to answer this question. I'm going to try to give a little context, but if you can't follow it, that's okay because this is now we're now going into a completely different genetic mechanism. So, what this person is talking about is PIK3CA mutations, and I study these a lot, and my paper really looked at these actually. So, the way that this works is, and I actually encourage you to tune out if this isn't something that you already are familiar with or know about, because it actually, like the reason why I didn't discuss this is because it adds a little complexity and confusion if you're not familiar with it. But basically, it's a 3-hit in reality, and I hate doing this change, but functionally, it's a 3-hit hypothesis where you need a mutation that makes these cells divide a little bit more, and that's PIK3CA. And then you need a mutation that destroys the function of the endothelial cells. So that's what I was talking about before. So I really focused on the mutations that destroyed the endothelial cells. So that's the 2 hits. The two, like you have the germline one, and then you have a second one. Now you have no CCM. That's the double hit. That's bad. But that alone actually won't cause a lesion. You also need those cells to grow a little bit faster. So that's where the PIK3CA comes in. In terms of which one happens first, we think, and a lot of the studies will show, that typically the PIK3CA happens before the second hit, and those actually work synergistically because when you have a PIK3CA mutation, those cells divide more, but they're also more prone to mutations. So you're actually increasing the likelihood of that second CCM hit. So what happens is the PIK3CA mutation happens, you already have one CCM mutation, then that cell divides because the PIK3CA is saying divide, and it's not dividing particularly well because when PIK3CA cells divide, they tend to get more mutations. And then as that process is happening, you get the second hit, and that's when the lesion grows. If you just have PIK3CA and one CCM mutation, lesion's not going to grow. If you just have two CCM mutations, lesion's not going to grow. You need both. And that's not, that's like we think that mechanism is the most common mechanism because people will have these things. This person, I'm sure, is familiar with it because the way they ask the question, it seems like they've read the literature. But you'll have these things called deep venous anomalies, DVAs, that precede the CCM. So you have a deep venous anomaly, which has a PIK3CA mutation, and then next to it, a CCM lesion grows. And because of that, it seems like the PIK3CA event is happening first, and then the CCM mutations are happening second. But in some samples that we have, it actually seems like clonally when we look at it, it seems like, but it's hard because there's such a small percentage of cells that noise is there, so we can't really confirm it. But in some cells, there's some evidence that maybe the CCM mutations happen first, and then that cell divides and gets a PIK3CA. We think the more common mechanism is PIK3CA, grows, another hit, big lesion. But in some instances, we do think that it's possible. And it's possible. And in fact, the more you do genetics, in my experience, the more the possible is probable. Basically, any genetic direction and mechanism you can think of happens. So I would be personally very shocked if sometimes it didn't happen CCMs first and then PIK3CA. But I think the predominant one is PIK3CA first, then the CCM ones. Okay, so I'm seeing these in the chat. The emotional stress cortisol release causes issues with CCM. So again, scientifically, stress is going to create like oxidative stress, inflammation, and oxidative stress and inflammation scientifically does tend to increase lesions. But again, like in terms of like what I think in general, stress is bad for everybody, and things that are bad for everybody, if you have CCMs, you're going to be more fragile. But it's like, what do you do to control stress? Though it's hard. So like, but yeah, like unfortunately, stress and like any sort of thing that's bad for most people is going to be extra bad for people with CCMs. How many times a CCM has bled and or risk of a rebleed, that is going to be a doctor question. So I don't really like. I'm not used to seeing these MRIs or these T2 scans, so I don't really know how to do that. So I know that like our collaborators will be like, okay, in these mice it's bled a lot, so that's like people at the University of Chicago, neurosurgeons, they know how to answer that. I don't know how to determine it, but I know that people do.
Yeah. All right. There's one question kind of way up high that might be, I think, a good question to maybe end on because we're just about out of time. So this question is, have you tried removing the sick endothelial cells? And if so, did new ever form?
Yeah, so when we try to remove the sick endothelial cells, so people are in the process of doing that now. And we're trying, like, typically what that means is gene therapy. So the way that we try to remove those is gene therapy. And gene therapy, we are in the process now of doing things to remove those sick cells and to actually just try to kill those bad ones. And those studies have been promising. But the thing with gene therapy is that you get off-target effects. It's really challenging and it's a long road. But there does seem to be early evidence, and this is very early, that when you get rid of the sick cells, it does help a little bit. Now, how much does it help? We have to see this in a lot of mice. We have to do it different ways. But it does seem like if you get some of the sick cells in a mouse so far, that does seem to discourage these lesions growing. But this is really early on in that, and I don't anticipate that gene therapy is going to be something that like is far enough along to do that. But that's something that we're really actively looking into. Like, how can we actually just target and hurt these cells directly? And a lot of people are now trying to work on this without gene therapy. In cancer, they do a lot of research, and we take a lot of our ideas from cancer. In cancer, they do a lot of work where they try to find something different about the cells other than the gene itself. So you don't have to change the gene, but you can find a way to recognize those cells, and then you can send your immune system to kill them. So people are also working on strategies like that. Are there different ways that we can kill these sick cells? But the field definitely thinks that if we can kill these sick cells, that there's so many healthy cells that you could be okay because some of these shrink. So we think that that really is a mechanism that could work, but we haven't yet proven it. We haven't gotten something far enough along to tell you 100%.
Got it. Is there anything else that you want to hit on or you think we should hear about tonight?
No, I think that's great. I think I would love people to email if they have questions or if they want to participate in simple science sessions about any sort of things in the future. Again, it's simplescience@alliancetocure.org. Is it .org or .com? I forget.
.org. I'll put it in the chat.
Yeah. Okay, beautiful. And then if I didn't answer any questions, great, you can email that too, and I'll follow up. And it's weird not seeing everyone's faces, but I hope you guys enjoyed it.
All right. I think that they definitely did. This is a super active chat tonight too, which is awesome. So thank you so much, Andrew. We really appreciate you. And I really appreciate how you can bring things down to a level that our patients can understand. So thank you for joining us tonight. We appreciate you.
Thank you guys for joining me. Have a good night.
You too. Everybody, I did put the link to the support group in the chat. So if you would like to join us for support group, it is there. I'm going to leave the Zoom open for just a couple minutes for you guys to grab that link, because once I close this Zoom, you will not be able to get the link from the chat. So grab it now and head on over there if you'd like to join us for support group. And I also put the email for the simple science sessions in the chat. So grab that also. And thank you for joining us tonight.